Okay, right. Thank you very much, and thank you for staying back on a little late, not very, not like Michelle said, not my fault. I will take 1 hour now. Right. So, no, I'll try to be very short and brief. So, this is the topic, and throughout the day, we are being listening this particular buzzword, artificial intelligence and machine learning. And this is specifically in last couple of months, in fact, 3-4 months back, because of Chat GPT and generative AI has become a very, very big buzzword. But here what I am going to talk about is the artificial intelligence-based particle characterization. Of course, it's not a generative AI. It's a supervised AI, machine learning model. And how we can use it for predictive analytics in the dissolution sciences. Okay, so I need, yeah. Oops. Yeah. So a brief about my company. I mean, we are into particle characterization and visualization tools, and we assist our partners to improve drug product knowledge and process understanding to affect better health outcomes. This is the new AI-driven automated particle analytics platform with novel enterprise solution software model, which can work on any generic microscope. Be it any microscope, you can just integrate with this. It's a cloud-based platform, SaaS platform, so easy to implement. And we have 2 offices. One is in Pune in India, the other one is in Princeton, USA. Yeah. So couple of customers who are already using not the AI part, but our traditional model, Teva, Abbott, Freshness, Kavi, Sanofi. But here in India, we have more than 100 customers, 125 customers, and all big names of pharma industry there. They are using our products. What we are actually doing is we are trying to integrate all the departments of pharmaceutical manufacturing side and R&D side, and we do particle morphology, particle classification in terms of API and excipient using this technology, particulate matter count, especially in the sterile dosages where you have USP 788, 789, 790 tests, hot stage microscopy, nanoparticle analysis, microbial colony counting and gram staining, smart proofreading. So I'm not getting into detail. We have a stall here, booth here. So tomorrow, whenever you have time, we can get into the more details about it. And all these processes are 21 CFR Part 11 compliant and Annex 11 compliant products. We are talking since morning, what is the role of particle characterization and visualization. So in pharmaceutical industry, particle size distribution, particle shape of active pharmaceutical ingredient is known to strongly affect the stability and the aesthetics of drug formulation. And the size and shape of particles used in pharmaceutical product can impact the dissolution rate and influence the solubility, adhesion, dispersion, and particles. That we all know. So what is the difference we are bringing in? So we all know that what is particle size, but we generally don't understand the role of particle shape. So here I'm just giving one example, because generally whenever you are doing the particle size analysis and trying to correlate your dissolution profile with the particle size, you are trying to see the particle size with the laser diffraction technique, which actually approximate every type of particle into equivalent sphere, and the diameter of that sphere is considered as the particle size. So if you have different type of particle shape, like spherical particle, cube, and cuboid of different side ratios, and if the volume of all of these particles are same, then you will get the diameter of, or the size of this particle as 10 micron in this case. But if you look at the surface area is very different. For the same size of particle, a spherical particle will have the least surface area, whereas it can go more than 45% if your particle shape is different. So why don't we check, why we check only the size? Why don't we check the shape of the particle when we are doing our analysis? And that is because there is no quick or technique available where I can get the particle shape. If I can get the particle shape along with the size, probably that will give more confidence when I'm doing the particle dissolution profiling. So for particle size is not the only information probably you will like to know. There are shape parameters which we call morphological details, like ferret's maximum diameter, ferret's minimum diameter, circular equivalent diameter, circularity, aspect ratio, convexity, and many more. So what we do is we have designed one technique, innovative product, which gives all this data. And with the help of all this data, we have done a case study, 2 case studies I'm just showing here, where there are 2 different particles where we got the laser diffraction similar particle size, but when we do the microscopic particle size analysis, number-based analysis, and length as the diameter, their D10, D50, D90 values are different, and so the dissolution profile. This is another experiment which we did. Mr. Yevle is here, and he has done this using our system. We calculated the particle size and shape data, and then we run the dissolution, and we for 3 different same API in 3 different recrystallization process, we get very different dissolution profiles. And this all correlation you can do very easily with this system. So what actually we have done is we have automated the microscopic analysis. So if you see the history of Nobel Prizes, most of the Nobel Prizes in the chemistry or physics, scientists have used microscope extensively. And that's how most of the discoveries are happening. So what we have tried to do is we have tried to empower this microscope so that it becomes a real tool for you, and you can generate so much of analytics out of it. So we convert every microscope into an imaging workstation. So you connect any make or model of microscope with our platform, it becomes an imaging station. And what it does, automatically you can get the particle details. It reduces your analysis time, reduces your development time, and of course the manufacturing time. But most importantly, it eliminates the inefficient, error-prone, and tedious analytical tasks. Now coming to AI/ML model, so I'm a little going quickly so that I don't want to make you wait longer. In AI/ML, again, what we have done is we have different types of modules. Over the years, with the help of our customers, we have created different modules. Now we have put all these modules in one AI platform or a cloud platform, which will do all this, which I talked about on particle morphology, classification, hot stage, nano, microbial analysis, which is CFU counting. So you can actually integrate all your departments into a single platform. How this artificial intelligence work? So as you are taking the images of the particles, you have prepared a sample, put under the microscope, you start getting the images. These images are, first level of images are scanned through the automated stage. So you prepare a sample, load, it automatically scans the entire area, and then checks, the machine learning engine will check whether this image is correct for analysis or not. And then classification, again, the second machine learning is used, where it takes the characteristics of particles and try to put into different buckets. And as you are using it, the database goes, become richer, and then artificial intelligence comes into picture, and it makes the system more and more intelligent. And you get all the fast, accurate, and reliable data. If I have time permits, we'll do a very quick live demo how it works. So this is the schematic diagram. It's a complex system, but I just tried to make in 5 steps. The first step is we are doing the image acquisition through microscope. The first machine learning engine will, as I said, decide whether this image is good for processing. Like there would be some images which are not in the focal plane, blur image, probably it will reject. There would be some images which has got too many agglomerations, you don't want to consider it for. So that's how the machine learning first step. After image pre-processing, the second machine learning does the classification. So whether it's an API particle or an excipient particle, so machine learning, how it works, I'll just show you. And all this data goes to statistical analysis, which goes into the artificial intelligence, and it becomes, as you are using it, it becomes more and more intelligent. So this is the first step of machine learning for particle classification. So as we took multiple images, there are all types of particles. The first is the visualization or algorithm. So in an image, it will identify all the particles, and then we apply the clustering algorithms. We talked about neural networks. There are multiple ways we do the machine learning, and what it does is that it gives the different clusters based on the various parameters. Now, this is a supervised machine learning, because the machine learning models which are available, they do some kind of classification, but here we are very much conscious about some of our own intelligence we want to put into it, because artificial intelligence is still artificial, so we have to put some real intelligence into it. So what it does is that it classifies all the particles, put the different clusters, and then we give the name to those custom clusters, like first is excipient or API, API 1, API 2 kind of thing. And that model you can train with different types of APIs. Once you start running it, it will capture all the characteristics and create the model. And when you run the formulation, it will actually pick up those API particles. Now you have trained your model. Now you run this classification into this classification model. It gets trained, and then you become, it is saved in your database as a structured model. Now, if you are running any unknown sample, so what it does is basically, through its characteristics, it will try to match with the available database, and then gives it label like this is API particle or excipient. So this is a basic, very simplified way of showing you this. So this is what we have now imagined with our system, MorphoViz, and I would request all of you to come here. We would be opening up the introductory licenses for the students who want to use this system. They can use it. So any kind of dosage forms, you prepare the sample, put under the microscope, it will capture all kinds of particles. Then MorphoViz platform, what it does is that it will calculate all the size details, D10, D50, D90. It will give the distribution graph, and then also it will give the morphology of each and every particle. And further to this, as we are discussing, and we would be trying to collaborate with the dissolution apparatus manufacturers, where we can integrate this platform, and then the first part we have already done, where we can predict which is this particle. We can, as you are running batch to batch, you can get the blend uniformity batch to batch, and also with the dissolution data coming in this you can actually predict the dissolution profile, whether it's pass or fail. So actually what we are trying to do is we are shaping the formulation quality from lab to line. So all this microscope, which experiment you are doing in the lab, can be just integrated to the production line. So this is our vision on which we are working. The first part was automation, which we have already done. So a normal optical microscope is now become an automated system where you can control all the parameters and ensure that you get the repeatability. It's not dependent on the person. Second part is integrating various microscopes in a lab on a single platform. That is also done. So through MorphoVis platform, if you have let's say 10 microscope in your company, all those companies come to this cloud platform. Next part is where you can actually connect all the departments of your company to a single platform. So all microscopes, all kinds of studies which you are doing can go into this platform. And going forward, we can use, we are already talking to some of the companies where we can integrate in their cloud or on the public cloud. Their all locations can be connected to a single platform. So anywhere you are running, either in India or United States, you are running a batch, you can get all the data at a single place. Which in turn, if all pharma companies decide we can connect all the pharma company to this platform, they can exchange the data and get the benefit whatever the other companies are doing the experiment. And in end, we can connect the entire healthcare ecosystem where all API manufacturers, excipient manufacturers, they can put their particles in this platform. Formulators can pick up those particles. They can run the predictive analytics of formulations before actually going into for the formulations. So that was a very quick presentation. I think, see, I wanted to run a live demo. Let us see if we are able to do it. Is it possible? Right. So this is the platform called MorphoVis. Now it's running live in the servers. So we log in. Now you can create a user and then you sign up. So this is how what you get is all the kinds of modules at a single platform. You can like IPV class, auto class, hot stage microscopy, CFU analysis, proofreading system, nanoparticle analysis, auto count, which is USB 788, 789, and all this is then user management, which is 21 CFR Part 11 compliant. So can we quickly see the P class, how it runs? So this is the P class, which is particle classification platform. So yes, so this is how whatever analysis you do, it comes into your database. We'll quickly show one analysis. Like this is how you have got all the particles which is uploaded, and then what it does is that it analyzes those particles and then identify every particle, and then it gives whether it's a based on the definition what we have put, whether it's a API or an excipient particle. So yeah, so it's little slow, but when you are into the actual scenario, you can actually it has done is that first of all it has identified all the particles, and the green ones are it has identified as a API particle, and the pink ones are excipient particles. So if you can just click on any particle, it will give all the morphological details like its size, shape, color, texture, and convexity, circularity, perimeter, width, all the details of every particle you can get. And then if you save them, and yeah, you can show that. So here you get the histogram of total. If you want to see the histogram of your API particles, you just click on API, you will get the histogram of API. If you click on excipient, it will give excipient. Here it is generating D10, D50, D90 plot for every image, and then the cumulative plot. So that what happens is it gives you the indication whether you have come to a stability. And then other details like D10, D50 D90 for API particles, for excipients, and for the total, and other particle details. So if we just...
This was a, This was a two-dimensional photograph, right, from the microscope.
Correct.
But how did he measure the third dimension?
So we'll just finish this and then I'll answer that. So we save the analysis, and you can just upload a number of images. No. Just save the analysis. Don't do it. So while saving this, it will give all other details, like what are the every image-wise details. Yeah. So it gives all other details, like I said, that minimum particle size, maximum particle size, average size, and then again histogram of all these details for every particle. Can you go below? Right. Here you get D1 to D100 all data. Whatever analysis we have done, once we click on this model training module, machine learning, whatever data it has given, it gives, let's say, in 3 buckets. These are the API particles, these are excipients, and these are, let's say, your agglomerations. What it does is that it gives you a supervisory feature. So if you feel that this is not my API particle, you can actually pick that particle and put into the agglomeration bucket, or some agglomeration which has been got into the excipient side, it goes into the... So here what it does is that it has got all the particles, and it gives all the morphological details of every particle, and you can click on any particle and it gives all the details of that particle. And if you want to see where this particle was when you are doing the analysis, you can click it and it will actually show that it was here. So that's how you can actually trace every particle when you are doing the analysis. Yeah. So there is one dissolution method where we are trying to correlate the particle analysis with the dissolution. So let's say you have done one analysis and then you have got all the particle. Now you can correlate your dissolution not only with particle size but all 14 different morphological details, and it will predict for, like, you can I've given a very simple example, but you can create more data into it. So let's say you are using USB type 2, quick, and then you can predict, you can put your dissolution, if you are connect with your dissolution apparatus, it will take all this data here, and then it will try to correlate, machine learning will correlate its data with the particle morphological details. So that is a nutshell. There are a couple of things, but I'll not get. So thank you very much for patiently listening to this talk.
Thank you, Sandeep, for this nice presentation. I know that you're trying to integrate your expertise in particle size measurement by microscopy with dissolution. Hope it will take a good shape and we get a more predictive and confirmed result very soon. A couple of questions from the audience, if there are any, because we are already running short of time. So if anybody has any question. Maybe tomorrow when you get time, you please visit his booth over here. He can demonstrate. Yeah, Sajeev has a question.
Yeah. What is the CFR compliance for your software? What is the level of compliance that you have?
So it's a 21 CFR Part 11 compliant fully. And it's, we have more than 300 installations in all the US FDA approved plants, so they have verified it. Yeah.
On the PMT test as per 788, 789 without automation?
Its semi-automatic version is there. Like USP 788 doesn't say that you have to use fully automated system. So this is a method 2. USP method 2, 788 method 2. There what you do is that you, if you are doing the semi-automatic, you pick up multiple regions. you know.
Manually.
So we know that for one image, how much area I have covered. So let's say I have covered 25% of the area. I extrapolate that value for the 100% filter paper area and then convert into the per ml or per vial particulate matter. That's also possible. But sometimes audit agencies are there saying that you are purposely missing some of the particulate matters. That's where a lot of companies go for the fully automatic system.
Okay, thank you.
Thank you so much. Thank you so much, our wonderful audience. Thank you, Sandeep sir, for those wonderful insights. And thank you, Rajiv sir, as well. But yes, please be at the center. Rajiv sir, could we please ask you to felicitate Mr. Sandeep Kulkarni? Ladies and gentlemen, please put your hands up together. We'd like to felicitate him with the memento and the bouquet.
Thank you.
I request all to kindly be with us. You have been such a sporty audience and you have been so patient with us all. But ladies and gentlemen, I am sure after every session we have the celebration and we don't want to miss it out. So please be seated, kindly be seated with us. We'll quickly go ahead with Utsav, a celebration of Indian culture. Ladies and gentlemen, we